Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Aulia Fadli, and today I would like to present our research regarding single nucleotide polymorphism and type 2 diabetes mellitus phenotypes association using gradient boosting. There are 4 authors of this research. First one is me, Aulia Fadli, from Computer Science Department, IPB University. Second one is Dr. Wisnu Anantakusuma, from Computer Science Department and Tropical Biopharmaceutical Research Center, IPB University. Third one is Ms. Lailan Sarina Hasibuan from Computer Science Department, IPB University. And the last is Mr. Yudi Herianto from Chemistry Department, Tropical Biopharmaceutical Research Center, IPB University. Now, let's move to our introduction. The development of human genome sequencing technology nowadays has led the medical field to a precision medicine era. Precision medicine is a medical field that aims to provide disease treatment according to a patient's genetic profile, environment, and lifestyle. Recently, precision medicine research is focusing on studying complex diseases such as diabetes mellitus or DM. DM has various patients, covering a wide range of ages, from thin person to obese person, and insulin deficient to insulin resistant person, with 90% of its patients as a type 2 DM patients or T2DM. Moreover, even with the same medication, every T2DM patient can have different response. For example, two people with the same glycemic control can experience different symptoms of diabetes complications, where one person has diabetic retinopathy and the other is not. The identification of patients' genetic information becomes one of the most important things in developing precision medicine for DM patients. These genetic information can be obtained by finding biological markers that can be used to identify the origin of a disease. One of the biological markers that we can use is single nucleotide polymorphism, or called SNP. SNP is the genetic variation of a single nucleotide in the DNA sequence and represents at least 90% of the variation in humans' DNA sequence. We can use association studies to find a relationship between a group of SNPs and phenotypes in a disease. The association is then verified to find out whether the group of SNPs obtained contributes directly to the phenotypes. There have been several studies regarding SNP association before. The first one, Lopez, in 2019, tried to find SNP importances to the result of the classification of T2DM disease patients using nanoforest. However, the study was not able to identify which SNPs had associations with T2DM directly. And then Tresnawati, in 2019, conducted association studies of SNP and T2DM phenotype using random forest to rank SNPs based on feature importances. Then the results are used to perform regression. Her result produced 301 SNPs associated with T2DM phenotype. But this result yielded too many SNPs, which could make validation tasks in the laboratory become expensive. Therefore, it is necessary to try another method in order to reduce SNPs which are associated with T2DM phenotype. One alternative method that we can use is gradient boosting. Caruana research in 2008 shows that gradient boosting method has better performance than random forest for data with dimension of up to 4,000 features. Gradient boosting can also find SNPs' importance on predicting phenotype values. This study aims at conducting SNPs association study with T2DM phenotype using gradient boosting. This method is expected to obtain fewer putative SNPs, which has comparable predictive ability than the previous method. Association study is conducted by ranking SNPs' importances, then selecting which SNPs considered as putative SNPs. These putative SNPs will be validated based on association data in Mouse Genome Informatics or MGA website and literature study. Our research begins with data acquisition. There are 3 data to be collected: protein candidates, phenotype data, and SNP data. For protein candidates, we use the same 98 protein candidates that used in Tresnawati research. Of all the 98 candidates, only 73 had SNP data. For phenotype data, we use the insulin tolerance data from 11 rats that were taken from Schoenfeld dataset from mouse panel database. These datasets contain 115 instances with 11 unique mouse strains and 21 features. For SNP data, we obtained it from mouse panel database 2 by querying the protein candidates we use with strain in phenotype data. There are 2060 instances or SNP and 16 features in SNP data. The last 10 features are mouse strain that matches the strain in the phenotype data, while the other 6 are chromosome location, display position, SNP ID, observed variation, protein annotation, and requested protein. After we get the data, the next step is data preprocess. For data preprocess, we have 4 stages. First is removing unused features. We remove all features in the phenotype data except strains and insulin features. We also remove all features in SNP data except SNP ID and its strain. The second stage is handling missing values. In phenotype data, we remove every instances that do not have insulin values. This process results makes phenotype data instances reduce from 115 instances to 95 instances. In SNP data, we remove all instances with missing values and H value and strains attribute. H value means that an element in particular strains can have a value of A or C or T. Therefore, to avoid ambiguity, we also delete SNPs with H value. In SNP data, the SNPs reduced from 2060 to 2032 SNPs. The third is SNP encoding using recode SNPs function in RStudio. Because of this function only accepts the allelic form SNP, we must transform SNP data to be an allelic form. We then encode SNP data with following condition. We transform into 1 if the allelic form SNP is major homozygous, 2 if heterozygous, and 3 if minor homozygous. The last step is merging phenotype and SNP data based on its similarity in the strain value. Merging results make the data has 95 observations, which is strains, with 2032 features or SNPs as predictor variables and insulin features as response variable for the model. The next step is building association model using gradient boosting. We use gradient boosting with best standard use decision tree and loss function least squares. We also use cross-validation to validate model performance with number of fault is 10. Article in No paper at 2013 suggests to use low interaction trees with max depth around 5 for gradient boosting with best standard decision tree, because this type of tree usually has good performance for real-world dataset. Therefore, we perform parameter tuning using grid.cve to find optimal parameter. There are 3 parameters to be explored: learning rate with value between 0.01 to 0.1 with 10 intervals, and estimators from 10 to 100 with 10 intervals, and max depth from 1 level to 20 level with 20 intervals. After the model produces the result, the next step is to do association analysis and evaluation. The analysis process is done by mapping the selected SNPs from our result to its genetic information. These association results then will be evaluated by validating the mapping results with the information of gene associations contained in the MGI and literature study. For the model evaluations, we calculate the minimum squared error or MSE and mean absolute error or MRE of the model. We now enter in result section. This is the result of the model. The initial model, the model before parameter tuning, produced an average MSE of 0.062 with standard deviation of 0.041 and average MAE of 0.17 with standard deviation of 0.049. This model also yields 254 SNPs selected. We then use grid search CV to find the optimal parameter of the model. The optimum parameter we obtain is learning rate, which value of 0.08, max depth is 1 level, and n estimators is 103. This model produces an average MSE of 0.061 with standard deviation of 0.04, MAE of 0.171 with standard deviation of 0.047. This model also yields 30 SNPs, which is much less than the first model. The feature importance value of these 30 SNPs can be seen from figure 1. We use these 30 SNPs to do association analysis. The top 15 SNPs mapping results can be seen in table 1. From this table, we get some interesting results. From the 30 SNPs result earlier, we get a total of 12 protein which has associations to these SNPs. The protein with the most SNPs is CDKL1, which associated with 9 SNPs result. We also find that the highest importance protein, which is CALN1 and FTO, who have the association with the most important SNP in this table, is the same protein which also have the highest importances in Tesnawati results. It means that the three methods recognize this protein to have some importance in predicting insulin values, but this result still needs more research. For evaluation result, we find that protein AKT2, CDKL1, IAPP, IGF1R, RS1 are directly associated with T2DM according to MGI and literature study. Meanwhile, FTO and INS are associated with one of the T2DM phenotype, which is insulin resistance. There are 22 SNPs associated with this protein, which indicate that these SNPs have an association with T2DM. We can see from evaluation results at table 2 that there are associations between SNPs and T2DM phenotype through SNP protein phenotype relationship. This result can be the basis of making a genetic profile to develop precision medicine for T2DM patients. For other protein information like protein FF3, CLN1, EPO, MPOS9, and RAT, we still have no information regarding its association to T2DM phenotypes on MGA website. So we tried to do a literature study and find some findings like CLN1 is associated to T2DM disease according to Gu et al. Research in 2019. But this study does not mention its function in detail. We also find that IFF3 and EPO have association with T1DM disease according to Regula et al. Research, but no information regarding its association to T2DM disease. For model evaluation, gradient boosting produced an average MSE value of 0.061 with a standard deviation of 0.04 and MAE value of 0.171 with standard deviation of 0.047 and also yield 30 SNPs. This result considered quite good for predicting phenotype values, and also compared to random forest method that used in Trustnawati research, which produced an MSE of 0.062 and yield 301 SNPs, gradient boosting method managed to produce much fewer SNPs with a slightly better MSE. So for the conclusion, gradient boosting produced 30 SNPs that have potential to have an association with the phenotype of T2DM disease. Its performance also quite good for predicting phenotype values. Information on MGI and iterative studies shows that 22 of the 30 selected SNPs are associated with the T2DM phenotype through the SNP protein phenotype relationship. In addition, the drawback of this study is the need to randomize the data before using grid search CV. This caused the result of the model to change if an experiment will be conducted in another programming language. Therefore, it is best to use another library for that. Our reference in our presentation. That's all our presentation. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh.